Okay, why do we measure RNA in white blood cells? Why do we use leukocytes? Because I've been telling you about muscle sensory neurons, right? So why are we using leukocytes then in our measurements? Because that's where we get our RNA from. Well, again, under the lamppost, we can obtain white blood cells easy. So it's easier to find your keys looking under the light. Well, it's not really, because your keys may be over there someplace, but and that's the truth. But it's much easier to look at it here. But there's more logic behind it than that. Brain and sensory neurons that innervate muscle don't grow back. Removing them is not advised, so we don't do that. Okay. On the other hand, white blood cells are also signaled by the chemicals made in your muscles, and we knew this beforehand. So we knew that white blood cells, they react to the signal that you get from your muscles, and so they're detecting the same fatigue signals that your brain is detecting. There is a tissue donation site in case you do want to donate your cells, and that's this particular thing. It's the NIDRI resource. I can hardly read it here. Sorry about that. NDRI resource organ donor programs, individual donors, and this is the site. If you just look up under tissue donation for sensory neurons, it's the National Donor Resource Information is what it is, or something like that. Yeah, I don't know what it stands for either. So, but they've actually been sending me emails on a consistent basis, telling me they have dorsal ganglion cells that I could buy from them, which are actually, and they're actually quite cheap, and they will provide them in a form that would be useful. Trouble, I can't use them right now because I don't know enough about how to use them yet. So, okay. So again, back to why do we measure RNA in white blood cells? Well, when signaled, the white blood cells make chemicals, cytokines, that cause sensory neurons to become more sensitive. We knew this. So we knew not only did they detect the fatigue, but in fact, they could actually be part of what's making your sensory neurons send more signals. And in fact, the white blood cells are the immune cell function cells, and they can increase fatigue through cytokines from a variety of diseases and trauma. We've known this for a long time. So even before you had chronic fatigue syndrome, when you got the flu, you felt fatigued and you felt muscle pain. Everybody does. And it's because the viruses cause the immune cells to react and make cytokines. Part of the things these cytokines do is they do help you tear up the viruses and other things, so they actually make you better too. But they also signal the sensory neurons that you're having fatigue and pain. So, and which is part of the reason, part of the idea is that it actually protects you. When you're sick, you want all your resources to be put into actually putting your immune system in action and not out running around and doing all kinds of things you normally do, because you want to save those resources to attack the disease. So that's, this is part of what white blood cells do. So part of a large group of people think that this may actually be the case in chronic fatigue syndrome, and that in fact the immune system may be instrumental in causing chronic fatigue syndrome. This has been a major hypothesis in the area for quite a while. So the white blood cells could be the primary cause of fatigue in some conditions. We actually believe, given what we've seen in terms of the activation of white blood cells with the exercise stress, that in fact they contribute not as much as many of the groups have thought. That while they can contribute, they're not a major contributor in a lot of these patients. Okay, so why does gene expression change in chronic fatigue syndrome and fibromyalgia? Why would it change at all? Why should it change? We don't know. That's the answer to that. Because there's not a strong genetic influence, not many strong family relationships, we believe that most of the causes of chronic fatigue syndrome and fibromyalgia are environmental. We actually have a collaborator who's worked with us that has found some families in which chronic fatigue syndrome seems to be inherited. Okay, we haven't been able to study that because we've been not able to get additional sources of fundings until just recently. She just recently got a grant, so hopefully we'll get more into that to try to figure out what's going on in that particular family. So some of you may know about Alzheimer's disease, for example. Alzheimer's disease, only a very small percentage of Alzheimer's disease is directly caused by genetic changes, something like less than 2%. Okay, but studying that 2% of people gave us clues as to what's actually causing the disease in all the other people. Okay, and that's what we're sort of hoping here. Even though we think that the familial relationship, the actual inherited chronic fatigue, is rare, if we could study them individually, we may come up with the clues of what's causing it in everybody else. So that's what we're trying to get at there. Okay, we do know that viruses cause gene expression changes in many types of tissues. So viruses are one of the possibilities for what could cause the changes in chronic fatigue syndrome and fibromyalgia syndrome. Particularly Epstein-Barr has been well studied and almost certainly does cause, maybe not all by itself, but at least itself and then something else, because there are many cases where Epstein-Barr virus mononucleosis was that is fatigue, causes fatigue all by itself, and these patients developed that and never got better. Okay, and they've been well studied. Could other kinds of viruses cause this? Most of you probably are aware there's a thing called XMR virus that's been really highly touted as maybe causing it. And as I told you at the very beginning, I'd like to say it did or didn't, and I can't tell you either. It's a very difficult virus to study. We have one of the world's experts right here in Salt Lake working with us to try to do this. Ela Singh, who was one of the people who first identified XMR virus as potentially associated with prostate cancer. And that's where a lot of the research initiated from her efforts. And she's an absolute expert in this area. Very, very careful researcher. I'm so glad we're working with her. She is just an expert. Okay. Injury and stress can cause gene expression changes, fairly dramatic changes. And toxins, allergens can cause gene expression changes. So it could be that any of the things that we think might be causing it are causing it. It's likely that some of them are, and some of them may be doing it in combination. So it may take a double hit, 2, 3. So it could be a virus and an environmental effect of another variety. So heat, cold stress, something else. We don't know yet. We need better models for this. We need to understand more what fatigue is, the final common pathway. What's really affecting the final common pathway there that's actually producing the symptoms? Not what's upstream, because everybody gets these upstream diseases and they all get better again. With chronic fatigue syndrome, you don't get better again. Okay, so now I'm going to go back to the beginning. I'm almost done. Which is, okay, so now I've got to explain what we measure and why we measure it. Let's go back to the findings again. Okay, the brief outline of the protocol. We do this blood draws, numerical ratings of mental and physical fatigue and pain, 0 to 100. Whole body air-dyne cycle, 70% to age predicted max exercise, 25 minutes. And then we have report all these things. We do post-exercise blood draws, 5, 8, 24, and 48 hours. That's the worst part of the whole protocol. I've done it myself as a control. Just getting all these blood draws, particularly when you're needlephobic like I am. It's amazing how many anesthesiologists are needlephobic. It's a real shocker. You'd be surprised. Then we give them all these tests, and if this doesn't fatigue you, nothing will. This actually made me more tired than I've been in quite a while trying to take all these tests. Okay. So we take all these measures, we do the gene expression levels on leukocytes relative to the housekeeping gene, and I showed you what all these receptors are. The sensory receptors, the alpha adrenergic receptors, the cytokine genes. So each of these are particularly, I explained them before, are particularly there because we knew these were sensory, that these were on the sympathetic neurons, and it turns out they're also on the sensory neurons, except even this is on sensory neurons. And then all of these we put on there because of the hypothesis the immune system was involved here. Of all of these, this is the only one. IL-10 was the only one we found was really changed in patients with chronic fatigue syndrome. Again, here's the data again, only a slightly different organization. Controls. Chronic fatigue syndrome and chronic fatigue syndrome patients that are comorbid for fibromyalgia syndrome. This is 34 of the patients right here. These are the ones without that alpha-2A decrease, so these are ones that did not go down. And here you can see if we put them together, this group looks even better than the data you saw before. And it looks better because this really is a relatively homogeneous group with the exception of this gene and that gene. Okay? So you can see how much greater this is. It's orders of magnitude greater than controls. This is the major subgroup once again. So this is the chronic fatigue syndrome and chronic fatigue syndrome comorbid with fibromyalgia syndrome. So all of those patients, initially I showed them broken into two different groups. It's all a single group here now. And now you can see that it looks even better than it did before because it's actually quite a consistent group if we average it all together. The genes that are different, this is the only gene that's really different here. The alpha-2A was down in this group. And again, 70% of these patients have orthostatic intolerance. This is fibromyalgia syndrome only patients, and here's the controls again. This gene, alpha 2A, is the difference. Okay, again, what about the fibromyalgia again? I showed you this before. Same thing again. Fibromyalgia only, no changes in controls. The changes are in these genes at baseline. Severity ratings, what were those? Well, this is what the severity ratings I showed you before were. With one being fully disabled, these were able to hold part-time jobs and do the majority of self-care activities. And here we go again. The highest gene changes occurred in patients with the highest severity, less so with less affected, less yet with those, and finally the least, with those that were the least affected. Controls. Okay, gene expression is biomarkers. It turns out these are controls right here. Now I just dot blotted them and showed the increases that occur from adding up these genes right here. And these are the most severely affected. These are less severely affected patients, and these are controls. And you can see that there's a pretty large separation here. And if you use just that with no real manipulations in it, you can come up with a biomarker that has a specificity of 94% and a sensitivity of 65%. This is not so good, the 65% right there. We think we can improve that by throwing in some other genes, but we haven't done that yet. This is actually quite good. The specificity of 94% is really good. If you put these two together, according to the gold standards of biomarker industry, this is a good, actually it's a good to very good biomarker. It's not an excellent biomarker, and we're hoping we can make it somewhat better. But this could be available, hopefully, in the not too distant future, as maybe 2-3 years, as something that could be clinically useful. Okay, so are biomarkers useful in treatment? Well, we'll start right here. The AE2A decreases, so we also has the orthostatic intolerance. This group should be treated differently, right there. So if you knew you were a patient that fell into this group, and if you had both orthostatic intolerance and this AE2A decrease, you're in that group, and you should be treated differently. Are the treatments helping? Well, the subjective measures can be misleading. Patients may be getting better but not know it, or they may be getting worse and not know it. Some patients may benefit, but which ones? Which ones would benefit? Well, it's very possible that the biomarkers that we're using, if we can look at it under these conditions, may actually help us determine these things. And we have a study that we're just starting, and hopefully it's going to begin any day now. We're looking at Lyrica. Pfizer's given us a free grant, and we don't have to give them anything for this, a grant to actually study a crossover design where we actually look at patients on Lyrica and patients that are on placebo, and then cross them over so that in the next period, the people who are on Lyrica will get placebo, and the other people will get the Lyrica, and they won't know what they've gotten. And we'll actually look at the gene expression and see whether the gene expression is getting better with treatment with Lyrica. Do existing treatments have effects on fatigue and pain? Well, yeah, we don't know that, actually. So this is data we had given the scale you just saw before for fatigue and pain. This is pregabalin or gabapentin or Lyrica, as the case may be. And this shows you that in our patient population, this is that same pain mid-exercise, pain after exercise, 48 hours after exercise, that in fact, the patients that were on pregabalin did better than patients that were on other treatments. But that doesn't mean anything all by itself, because this was just taking patients that were on it, and they may have been on it for the reason that they're doing better and all that kind of thing. So we need to actually do the real study, but it's possible. Mental fatigue, same thing.